So our next speaker is Shermaine Sy. Shermaine Sy is here in Edmonton, and she is a trainee from Dr. Laurie West's lab at the University of Alberta. And she's going to be telling us today about the development of a bead assay for ABO antibody detection. So without further ado, Shermaine, the floor is yours.
Hello, everyone. My name is Shermaine, and I'm a recent graduate from the University of Alberta Medical Laboratory Science program. Today, I'll be talking to you about the research project that I did about utilizing Luminex Technology to study the development of anti-ABO antibodies in pediatric patients. So just a bit of background information. The ABO blood group system consists of 4 blood groups, A, B, AB, and O. The ABH genes will encode for glycosyl transferases that add carbohydrate units to form the carbohydrate antigens. Anti-ABO antibodies are reported to develop at 6 to 12 months. Lack of these preformed antibodies to ABO is an opportunity for transplant. Once patients form antibodies, transplant is restricted to ABO-compatible transplant. Otherwise, this can result in hyperacute rejection. From previous studies, there are at least 4 subtype structures that are expressed in humans, and the expression varies amongst tissue and cells. So for example, group A red blood cells are known to express subtypes 2, 3, and 4, while group B red cells and the heart express subtype 2. This is very important for you to know because red blood cells are used in hemagglutination, which is what is currently used by the clinical lab to measure anti-ABO antibodies in pre-transplant specimens. Now here, shown on the right, is what a hemagglutination assay would look like on a 96-well plate. Hemagglutination is when clumps of red blood cells form from A or B antigen on the red cell surface, interacting with anti-A or anti-B. These results can be reported qualitatively as positive or negative for agglutination, or quantitatively as a titer, which is the highest dilution that causes agglutination. Hemagglutination for anti-ABO antibody detection has many limitations though. First off, it's not standardized, and there's a lot of variability in the choice of platform by each lab, as well as the technique and the interpretation by each technologist who is running the test. It also has poor reproducibility and detects predominantly IgM antibodies. And lastly, it lacks ABH antigen subtype specificity to account for differences in the tissue versus the red cell ABH glycan expression, which is very important when you're determining if a patient is suitable for transplant. So in order to overcome these limitations, the West Lab, in collaboration with the University of Alberta carbohydrate chemists, Dr. Todd Lowry and Dr. Chris Cairo, created a solid phase assay that uses Luminex technology. Now, to briefly describe Luminex technology, it uses polystyrene beads that are labeled with different proportions of red and infrared fluorophores, thus producing 100 unique beads that can be identified using Luminex detection systems, such as the Luminex 200 cytometer. Conjugation of a specific capture molecule, such as an antigen or an antibody, to the beads allows for detection and quantitation of the analytes in the sample. So to help visualize how this works in our assay, this is our ABO subtype 2 bead. And any anti-A2 antibodies that are present in the patient's serum will bind during the incubation. And then we can add our phycoerythrin or PE labeled secondary antibody that we can use to detect any bound patient antibody. But first, to prepare the Luminex beads for our assay, we use a Premade Kit that has our reagents to couple bovine serum albumin-linked ABH subtype antigens to the Luminex beads. And then in order to ensure that the coupling was successful, we use fluorescently labeled monoclonal antibodies to ABH subtype antigens. Moving on, the aims of our study were to utilize the Luminex bead-based panel that was developed by Dr. Laurie West's research group to measure antibodies to ABH subtype antigens. We also wanted to investigate the age at which IgG or IgM antibodies develop and to confirm and perhaps expand the age window for ABO-incompatible heart transplants. Now moving on to our methods, our study consisted of 31 heart transplant patients who had pre-transplant serum samples that were less than or equal to 24 months of age. Patients had multiple samples, which means we had 86 samples for Luminex analysis, and we were also able to run hemoglobination in parallel on 55 samples that had enough volume. Next, for our Luminex bead assay, we first prepared dilutions of our patient serum, and then we can use a 96-well plate to add the beads and then the patient serum, and we have separate wells to detect both IgG and IgM, anti-A and anti-B subtypes. And then we'll incubate this plate for 30 minutes at room temperature, and then wash it with buffer 3 times to remove any of the unbound anti-ABO antibody. And then we can add our IgG and IgM secondary PE labeled antibody that will bind to any bound anti-ABO antibody. Then we have another incubation wash step, and then finally the beads can be acquired on the Luminex 200 cytometer, and the results are reported as the mean fluorescence intensity, or MFI for short. For our hemagglutination, we first prepared our reagent A and reagent B red blood cells, and then again, we can use a 96-well plate to prepare our serum samples by performing twofold serial dilutions from 1/1 to 1/2048. And then we add our reagent cells to separate wells and incubate for an hour at room temperature. And then the results can be read, and the titer is reported as the highest dilution that causes agglutination. Moving on to our results, we had 86 samples available, 40 of which were group O and 46 were group A. We had 1 group B patient, but they were excluded since they only had 1 sample, and we had no AB. The median ages of the samples were 8.2 months for group O and 9.9 months for group A. Several of our patients did receive blood products or transfusions, which is a limitation in our study since it may have affected both our Luminex and hemagglutination results, but we're still collecting this data from our patients and from the transfusion records, so it is not yet being considered in the figures I'm about to show you. So the next 2 slides will show the anti-ABO antibody development over time. So please note that these patients had multiple samples at different time points, so they're all represented on these graphs. So this first slide shows you the anti-A2 and anti-B2, IgG and IgM for group O patients. I'm only showing you the anti-ABO subtype 2 reactivity because this is the target in the heart. And as you can see from these different scales, the anti-A2 was higher than the anti-B2 for IgG, which suggests that the group O patients may be at a lower risk than an A patient if they receive an ABO incompatible transplant from a B patient. We also see in that some of our younger samples, there was high IgG MFI, which could be passively acquired maternal antibody, but we don't know this for sure because we did not have the maternal data available to us, and also it wasn't observed in all of our younger samples. The IgM, we also don't see much of a pattern between the IgM and the IgG. In some cases, the IgM does appear higher than the IgG, but we're unable to determine if these were due to transfusions or if the patient just started making antibodies at that point. Next, I'm comparing our group O patients and our group A patients by looking at the anti-B2. So we observed that the anti-B2 for IgG and IgM were higher in the O patients than the A patients, which suggests that A patients may be at a lower risk than an O patient when receiving an ABO incompatible heart transplant from a B patient. On this slide, I'm showing you the IgG and IgM MFIs versus the titer that was obtained from hemagglutination. The clinical lab currently uses hemagglutination to assess risk in transplant, so this is why I want to compare these results to Luminex. And again, to remind everyone, hemagglutination uses red blood cells, so group A expresses subtypes 2, 3, and 4, and group B expresses subtypes 2. So this is why we're looking at the reactivity of these antibodies. Overall, we observed no correlation between the 2 results, which kind of makes us wonder what is driving these titers that we see here. Some possibilities could be antibodies to non-ABO antigens, which are usually rolled out in transfusion medicine lab by a screen that is done in the same conditions as our hemogglutination. So it's very possible that they may be contributing to the patterns that we see here. This observation is very important because in transplant, when you're looking only at the hemogglutination data, you could falsely say that a patient is not suitable for transplant, but really they have very little antibody. So to summarize our results, the group O patients, anti-A2 was higher than the anti-B2 for IgG, which suggests that they may be at a lower risk if they received from an ABO-B than ABO-A. We also observed that the anti-B2 IgG and IgM were higher in the O patients than the A patients, which suggests that A patients may be at a lower risk than O patient when receiving from an ABO-B. And lastly, the hemoglobination data highlights that red blood cells, which are used in this assay, do not express the same subtypes as the heart, as there was no correlation between the MFI and the titers. So now we'll move on to a case study. I intentionally selected a patient that had a complex case to highlight some of the issues of transfusion in young cardiac patients. So this is patient 433. They are an ABO male who was born in 2008. They had a diagnosis of left ventricular non-compaction cardiomyopathy and received a mechanical heart pump while waiting for transplant. At 15 months, they were transplanted and the donor was group A, and they had no cellular or antibody-mediated rejection 1 year post-transplant and no cardiac allograft vasculopathy as of September 2015. Now, although he's been transplanted, I wanted to show you some of the pre-transplant data because it's important to decision-making. So for this patient, we had 6 samples available from around 2 months to 14.5 months, and they received blood products prior to their last 4 samples, shown here with the blood drops. Now this patient is ABO-O, so we graphed their IgG and IgM, anti-A2 and anti-B2 over time. Looking at their 5th sample, they received a lot of blood products here, particularly albumin, in comparison to the 6th sample, where they received less product. However, the MFI appeared a lot higher in this sample versus the 5th sample. A possible explanation for these results could be that the blood products may have, quote unquote, diluted the antibodies in the 5th sample. Now, looking at the 2nd and 6th samples, we see that the IgM and anti-A and anti-B were high, but they weren't high enough to drive bead-based, thus producing a titer of 0 or 1/1. However, in the 5th sample, we observed 2 things. First, there was less IgM in comparison to the 6th and 5th samples, but still caused agglutination. And second, although we had more anti-A2 than anti-B2 by MFI, they both produced a titer of 1/8. So some possible explanations for these results could be non-subtype 2 antibodies, or even the high amounts of albumin that was administered that could drive agglutination here. These data highlight that patients, when they have a history of transfusion, the results need to be interpreted with caution. Moving on, as I mentioned earlier, we had some limitations in our study. In our younger patients, we observed the high levels of IgG, and we know that IgG can cross the placenta, but information regarding the maternal blood type wasn't available to us, so we can't conclude if these are of maternal origin or if the patient just started making IgG antibodies very early on. Oftentimes, when you're doing research on clinical specimens, you have to be mindful of when the samples were collected and if the patients were administered any products that could affect our analysis. Again, we're still collecting this data for all of our patients from our clinical transfusion records. And lastly, we had a fairly small sample size since we're looking specifically at pediatric heart transplant patients. So some of the conclusions that we can make are hemoglobination results are not accurate in the context of transplant, and Luminex may provide a better risk assessment. Our data that uses more sensitive and specific methods confirm a similar time frame for anti-ABO antibody development, but that IgG and IgM antibody development is not consistent between patients. And lastly, maternal antibodies and blood products should be considered in interpretation of the results. Future studies can collect more variety in blood groups to achieve a normal distribution. We also didn't get a chance to examine sex differences in our study, so that might be interesting as well. And lastly, we could also study the antibody development in pediatric patients post-transplant using Luminex technology. And the last thing I want to say is thank you to the members of the West Lab, and Halpin, Dr. West, Dr. Matika, Dr. Meyer, Jean Piercy, Tess Ellis, Ingrid Larson, Morgan Sosniak, Janet Zhao, and our collaborators, Dr. Cairo, Dr. Lowry, Dr. Ditchk, Dr. Campbell, Dr. Urschel, and the HLA laboratory staff from the University of Alberta Hospital for helping me with this project and also putting together this presentation. And also thank you to our patients and volunteers who provided their samples. And also thank you to GlycoNet for having me. And I'm happy to take any questions now.